The circulatory system, the respiratory system, and all the things involved with transporting oxygen to the mitochondria and CO2 back out to the environment are only one side of the metabolism coin. These systems all supply the oxidants to the reactions that release and tap energy from chemical fuels. Those chemical fuels, which we also call food, are the other side of the metabolism coin. Whereas oxygen delivery is handled by the cardiovascular and respiratory systems, fuel delivery is handled, at least at first, by another interface with the environment, the digestive system. Let's begin by talking about the nature of food. We've already covered some of this when we spoke about metabolism and oxygen. Broadly defined, a food is any substance that can contribute electrons at high potential energy and donate them to compounds with low potential energy, also called waste. The difference in the electrons' energy as they make this migration can potentially be captured as free energy to do work. Glucose metabolism, for example, involves taking electrons that are residing in the complex molecule of glucose and transferring them through a series of reactions that place the electrons ultimately in bonds in water. Glucose acts as an effective fuel because it can contribute electrons living in covalent bonds that it took a great deal of energy to form, and shunts them to lower energy covalent bonds in water. The energy lost in the transfer has not gone away. The first law still applies, but by channeling the electrons in just the right way, the energy can be used to do chemical and physical work, usually through the production of ATP. Unlike prokaryotes, which can use anything from rocks and strong acids to hydrogen sulfide as foods, or plants, which can manufacture their own food from sunlight, carbon dioxide, and water, animals are obligate heterotrophs. Furthermore, they are heterotrophs that aren't very versatile in the kinds of molecules they can use as foods. These fall into three broad categories: carbohydrates, lipids, and proteins. Let's turn first to the carbohydrates. The basic formula of a carbohydrate literally ties carbon and water, carbohydrate, together into a single molecule known as a monosaccharide. The most common carbohydrate fuel is glucose, which is known generically as a hexose. This means that the N in the generic formula to the left is 6, which makes the chemical formula for glucose C6H12O6, as we've seen many times before. Carbohydrates can, however, occur as pentoses, heptoses, or other odd combinations, but for various reasons, hexoses are the norm, at least for the metabolic fuels for animals. Carbohydrates are very versatile molecules. One form of their versatility is the ability to form stereoisomers of the same molecule. The most common form of glucose, alpha glucose, looks something like this. And if you want a more detailed stereo picture of these molecules, of course, you can always look them up on Wikipedia. For our purposes, glucose occurs commonly as a six-sided ring, in which one of the vertices is occupied by oxygen and the rest are occupied by carbons. Glucose can also open up into a linear molecule, but we'll stick with the ring form for now. The carbons are conventionally numbered 1 through 6, and the sixth carbon juts off in a position above the ring. You can see that the various hydroxyl groups attached to the carbons at the vertices are positioned above and below the ring. Among glucose molecules, alpha glucose is one stereoisomer in which the hydroxyl group is attached below the 1 carbon, on the opposite side of the ring as the 6 carbon, and on the same side of the ring as the hydroxyl attached to the 4 carbon. Glucose can also exist as a beta form in which the hydroxyl attached to the 1 carbon sits on the same side of the ring as the 6 carbon. We'll be coming back to this form of glucose later. Besides the alpha and beta forms of glucose, other hexoses are possible in which the locations of the hydroxyl groups with respect to the ring can vary. If you count up the number of possibilities, there should be 16 different stereoisomers of glucose. Don't worry, we won't go through them all, but we do have to look at some though. For example, an important isomer of glucose is beta-galactose, in which the hydroxyl attached to the 4-carbon sits on the same side of the ring as the 6-carbon. Finally, hexoses don't need to exist as 6-sided rings as is the case with glucose, but they can form into 5-sided rings. An important example of this is fructose, or fruit sugar, shown here. The temptation is to call this a pentose because it has this 5-sided ring, but don't yield to the temptation. It's a hexose with the chemical formula C6H12O6, just as glucose or galactose. A pentose would be a 5-carbon carbohydrate, C5H10O5. Another thing that makes carbohydrates so versatile is that they can polymerize. The simplest type of glucose polymer combines 2 sugar molecules into a single so-called disaccharide. The 2 molecules combine through a type of reaction called a dehydration. Let's look at the reaction for joining 2 alpha glucose molecules. Dehydration involves stripping away a hydroxyl and a hydrogen from the 1 carbon of one glucose molecule and the 4 carbon of another, which combine into a water molecule. This is where the term dehydration comes from. The 2 hexose molecules are therefore joined with a so-called glycosidic linkage. Because the linkage involves 2 alpha hydroxyls, this is known as an alpha glycosidic linkage. Dehydration reactions can occur between any of the hydroxyl and hydrogen groups on different monosaccharides. So, for example, we can combine a beta-glucose on the left with an alpha-glucose on the right. The dehydration takes away a hydrogen and a hydroxyl group from the 1 and 4 carbons of the respective glucose molecules, forming a glycosidic linkage. Because the linkage is now between an alpha hydroxyl and a beta hydroxyl, this is known as a beta glycosidic linkage. Keep these 2 types of glycosidic linkages in mind because they will be important when we talk about the digestibility of polysaccharides. There are some common disaccharides that we need to know about. The first combines galactose and glucose with a dehydration reaction that forms the disaccharide lactose, or milk sugar. As the name implies, lactose is an important dietary constituent of milk. When lactose is digested, the synthesis reaction is reversed by an enzyme known as lactase. This reaction splits the glycosidic linkage by adding the water back, a reaction known as hydrolysis, which literally means water splitting. Because lactose is such an important dietary sugar, most mammals produce lactase throughout their infancy. In most mammals, once the young are weaned, the lactase gene is typically switched off, and with it the ability to digest lactose. This is often the case for humans too, but in many human cultures, dairy products are used throughout adulthood. In these cultures, the lactase gene is not switched off after weaning, but continues to be expressed through adulthood. Sometimes this doesn't happen though, in which case the person loses the ability to digest lactose. And this leads to an uncomfortable syndrome known as lactose intolerance. We'll have more to say about lactose intolerance a little bit later. Another important disaccharide is sucrose, or what we know as table sugar. Sucrose is commonly produced in plants, with sugar beets and sugarcane being the notable examples here. Sucrose is formed by combining glucose and fructose through a glycosidic linkage, again through a dehydration reaction. This ability to join monosaccharides through glycosidic linkages can be carried on indefinitely, which means that monosaccharides can polymerize into much larger molecules called polysaccharides. One of the most important polysaccharides for the purposes of nutrition is called amylose, also known as starch, which consists of an open-ended number of alpha-glucose molecules stitched together through alpha-glycosidic linkages into a long-chain polymer. Molecules like starch form a class of polysaccharides known as storage polysaccharides. They're called this because they're used as a temporary depot for the storage of excess glucose, either produced by photosynthesis or in animals as an aftermath of a sugar-rich meal. In green plants, photosynthetic production of glucose that is in excess of the plant's immediate needs is combined into starch molecules and stored away. When the need for glucose exceeds the ability of photosynthesis to provide it, molecules of glucose can be cleaved off the polymer one by one and fed into the plant's own metabolism. When animals eat plants, of course, they do the same thing with the ingested starch. Starch is digested by cleaving off glucose molecules one by one for absorption by the intestine. We'll come back to this process a little bit later. Plants also produce another important polysaccharide, but in this instance, it's not used to store glucose for later metabolic use. Rather, it's used to provide structural integrity to the plant. These are called structural polysaccharides. In this instance, glucose molecules produced by photosynthesis are combined into a polysaccharide with the glucose molecules joined by beta-glycosidic linkages. This forms a long fiber called cellulose. Cellulose is an important structural component of wood. When numerous cellulose fibers are glued together with a protein glue, generically called lignin, it makes wood. Animals also synthesize storage polysaccharides, but these are not in the form of starch. The storage polysaccharide of animals is known as glycogen. This consists of linear chains of glucose molecules, but also joins other linear chains at various branch points. Glycogen is more than simply a chain of carbohydrates, though. It's synthesized around a protein core known as glycogenin. Numerous chains of glycogens nucleate around this protein core and are built up to form a glycogen granule. This represents a very dense depot of stored glucose, and it's synthesized during periods of excess sugar intake that commonly occur following meals. As in plants, when metabolic demand for glucose exceeds the supply from glucose dietary intake, glycogen stores can be tapped to release glucose molecules one by one, which can then be fed into the animal's own metabolism. Animals use carbohydrates to make their own unique structural polysaccharides as well. Because these aren't usually used for nutrition, we won't dwell on them here, except to say that they are based around a modified glucose known as an aminoglucose. What distinguishes these molecules from ordinary glucose molecules is a so-called peptide linkage through the number 2 carbon. In addition, many of the mobile hydrogens on the hydroxyl groups are replaced by so-called R groups, which can include things like sulfur or other complex carbohydrate molecules. This makes them a very diverse type of sugar, and when they polymerize, they form a class of structural polysaccharides known generically as mucopolysaccharides. They're also known as Glycosaminoglycans. These make up the basic structural foundation of things like cartilage or complex fluids like mucus or the synovial fluids that are found within joints. These compounds have a general springiness to them that makes them well suited for tissues that have to act as shock absorbers or lubricants. Carbohydrates are only one class of chemical compounds that can serve as foods, that is, as sources of energy, and materials such as carbon, oxygen, and hydrogen. They're also the only fuels that can be metabolized anaerobically, that is, in the absence of oxygen, largely because they are the only class of food molecules that can feed into metabolism via the glycolytic pathway. Animals also use other molecules as fuels, though, with the most common being the next class we will consider, namely lipids.